Finished this internship at the end of August last year. It's my first time going over all the vlogs, and I was hoping to find some sort of storyline that was going through it, but I'm not gonna lie, I don't really think there is one. So I'm just gonna put this out there, some messy snapshots of the work I was doing when I was at Microsoft, and hopefully it's insightful in some way. And I think it's probably a fairly accurate representation of the research process, which is just a bit all over the place. You're kind of with me on that journey as I'm figuring out some of the challenges that I'm dealing with, figuring out some of the modeling choices that we're going to make, etc. And yeah, there aren't too many vlogs anyway, there's only 5 of them, and I've made some interesting camera angles at some point. There's one where you're kind of up in my grill a little bit. There's an interesting lighting choice I made in one of the videos, and I also did a vlog outside, and so the wind was going a bit crazy in that one. But hey ho, my whole thing was just, if I remember that I got a camera with me, pull it out and record, simple as that. So that's kind of what you're seeing. Bit of context about the project. I was working on proteomics data for Parkinson's disease, which is basically, we had like a bunch of proteins. We're trying to figure out which drive the progression of Parkinson's, which is a well-known neurodegenerative condition. And I did this in the BioML team at Microsoft Research in Boston. So I'm gonna just drop you straight into week 1. I hope you find it interesting or insightful, and please tell me if you do, and also if you don't. Great. Reflections on Microsoft week 1, what we've been up to, getting the data, getting a bit of understanding about, for my project, Parkinson's disease. Had a chat today, like my first meeting with all of the other 3 supervisors that I have, so Kristen, Lauren and in the biomedical machine learning group. That was great, just to kind of get I guess like a semi sort of roadmap of where we're gonna go for the project. But you know, this is a thing I've understood more and more as I've done research over the past few years, is that it's just so much messier. It's nice to have a plan and like a final aim for what you want to do, but exactly what that path will be when you get there, very unpredictable. This is week 2 now. I'm kind of getting more into the swing of things, understand the problem a little bit better. Basically what we're trying to do is, given a clinical time series, predict it from baseline with another modality. And so I'll go into a little bit of what that means. So my supervisor, Kristen, a model that she developed before for Parkinson's was a clinical time series model. So given clinical features over time, namely motor skills, so like how well they can move, as well as non-motor skills. So that's things like depression or anxiety, that kind of thing, cognitive metrics. What they were trying to do was predict how the trajectories evolve over time. And the aim was that instead of kind of having these static subtypes at baseline, which is the first time point that patients have to stick in as the disease develops over time, what they can do instead is like have these stages over time. And so it's a more dynamic way of having subgroups, basically, was the aim of doing it over time there. And my aim now is, we have proteomics data at baseline, so that's protein data. We have all these proteins, now can we predict how they relate to each of those stages? And so the point there is that, like, if you can find certain proteins that are linked to each stage, you've done what we call biomarker discovery, which machine learning terms, that's a feature selection problem. Here it's a bit more related to kind of, can we understand like an underlying biological process for each of these clinical features? So for example, maybe there's a stage where you have very high rates of depression and you have certain clinical characteristics of motor characteristics like bradykinesia, so that's like slowness of movement. Maybe depression and bradykinesia are super high, and you figure out the proteins A, B, and C are involved. So is there some kind of biological process underlying those that would explain why the depression and the bradykinesia are flared up? And that's kind of the incentive behind what we're doing, which is, I think, a pretty cool problem. So yeah, hopefully we're gonna work our way through that a little bit better throughout this. I'm still kind of fiddling around with the data, so maybe there'll be an update on that one next. Cool. Just going through the data a bit more and kind of trying to understand it a bit better. And what we basically have is proteomics data from a platform, an assay developer called Olink. And the way that we've got it, the way that Olink do it, is that you get a set of proteins within panels based on some kind of mechanism. So you have like an inflammatory panel, which has all the proteins related to inflammatory processes, and then you have one for neurology, you have one for cardiometabolism, and then you have one other one which is just escaping me at the moment. But you have 4 panels basically, and the aim is to kind of standardize across them And they normally have a few proteins in common across the panels in order to help that. But another spanner in the works is that we also have different fluids. So for each, we have cerebrospinal fluid, which is like right near the stuff that we care about that's going on in the brain. Then you also have blood plasma, which is another thing that you can take, which is more helpful because it's non-invasive, but less likely to have as strong a signal, we think. So we have the same proteins, we have the same 4 panels, but we have them for both fluids, which is a bit strange as well. And what the PD, the Parkinson's people, the experts, were saying to my supervisor, Kristen, was that basically we don't expect there to be any link. So we should basically treat them as separate things. And I don't know if that makes it simpler in that like you just don't even consider them together, or if that means you treat it as a multimodal problem instead. So there's that whole question. I mean, I'm thinking multimodal because like having common features seems like a very useful thing if you can figure out how to encode it in the right way into the model. And then the final thing is that you also have different plates. So for some reason, all of the patient on like one of the experiments that Olink have done, and I think that's basically what a plate corresponds to, is like an experiment. And so it's a bit of a weird problem because what you're having to do is standardize across not just panels, but also fluids and plates. And I tried one really simple thing, which seems like it works, which is just to kind of like subtract the means. So I did a PCA plot of like the ones from either plate, and they looked kind of different, and I was a bit worried. I was like, how am I going to normalize across this? But then if you just kind of take the mean of one of them and subtract it off the other, it seems to kind of get the PCA plots in some kind of regular looking order. But then, you know, still kind of iffy in terms of when I run it through like more standard machine learning models like a random forest, it's not really coming up with much signal based on like predicting the Parkinson's people from the controls. I'm not too sure on like the best way to proceed beyond that, but I mean, it could also be that just the kind of data we have doesn't really tell you too much about Parkinson's versus controls, and it's more like within just a Parkinson's cohort that it would be better at differentiating. Those are some of the issues that I've been dealing with at the moment. The other kind of major thing is that it's a very small data set, right? So it's like only 200 odd people or something like that, 220. I think we're maybe expecting some more, but we'll see. The only way that you can deal with such a small data set from a clinical trial that has such a high number of features, like 1,500 proteins or whatever, so like 300 per, 250 per panel, about 1,500 total, is that you have to encode the model with some kind of prior knowledge. Kristen and my other supervisor, Lauren, have been working on this idea called GP bands, which is basically like, so the GP stuff, I'm not fully sure how it works exactly yet, but it's like a sample level Gaussian process where the hyperparameters are learned in a shared way, I think. Learned in a shared, with a shared neural network. And we want to make that neural network a biologically annotated neural network. In other words, an interpretable neural network that my supervisor Lauren developed. And so that's kind of what I'm thinking about ideation wise. I've tried like a few simple models and it hasn't really gotten me too far for like predicting Parkinson's outcomes, but I mean, maybe it will be better when we're actually predicting the clinical progression trajectories. Who knows? Let's see. My supervisor Lauren has this paper called Biologically Annotated Neural Networks, and the whole point of it is that you have sparse connections, or you have specific types of connections in the neural network. It's not fully connected, that reflect some sort of biology. And then that makes sense. That's something that's been done by other people, but the actual underlying algorithm that optimizes this whole thing is one based on a combination of importance sampling and mean field variational inference. Don't really need to worry too much about what they mean, but it's essentially that you have some approximating distribution, you have the actual distribution of your data, and you want to make that gap as small as possible, so your approximating distribution is as similar as possible to your actual data distribution. Now, the point of having an approximate distribution is that it's a simpler distribution that's easier to actually optimize over, and it makes it a tractable learning problem, which basically just means you can do it. You can actually train the model. And the way that it was done, like normally from traditional variational inference literature, is that you have loads of different, you have your loss function, which is like the elbow, the evidence lower bound. And then you take the gradients of that, partial gradients of that with the parameters that you care about optimizing, and then you just manually do those gradient updates. What a paper that was produced, I think it might even be a decade old now, but it's like, it's called black box variational inference, where essentially you don't need to manually calculate the gradients yourself and then do the manual updates. What you can do instead is take more of a deep learning approach And as long as you set up the loss properly and the parameters in your actual neural network instantiation, then you can train a model without having to calculate the gradients, because you just know the parameters you have to tweak, and you let like the way that standard neural networks are trained, which is stochastic gradient descent, or some version of like Adam W, whatever, and you just let that kind of figure out your parameter updates. And I hadn't really put that together, that is what we were going for. I didn't even know it well enough to even articulate the question, but I was going over this with my supervisor Kristen yesterday. We were writing in stuff up on the board, and then it just kind of clicked to me. So I've been basically doing Bayesian neural networks now for the past day or two, and then looking at making the connections for that sparser, looking at how then to get a more useful prior, which is the spike and slab prior. Essentially, a spike and slab prior is like, it's either nothing or it's a slab distribution, which in our case will be a normal distribution. And that's on each of the weights. So you have like sparse Bayesian neural network with spike and slab priors over all the weights. And the other thing was that like I was just way more proactive about it this time around. This week I've just been, more ad hoc, like messaging Lauren and Kristen about just like questions that I had, and we just sorted them out quickly. And I think about like, if I hadn't done that, how long would it have taken me to understand this thing? And would it have actually been worth that whole experience of learning? Because sometimes it is. Sometimes it's worth going through the hard thing yourself. But other times it helps to just have that little bit of support, or that big bit of support, maybe in my case, so a few times, just to kind of get you to that end goal earlier. It's just like this perpetual problem of like, should I be learning it hardcore myself, or should I, like, what are the okay things for them to help me out with? And I'm kind of leaning more towards, well, this is a short internship, just make use of the people around you, make use of the resources that you have. The project's going pretty well. Like, I finally managed to figure out how to get this specific type of prior working for a Bayesian neural network. And I also kind of realized that this paper that my supervisor Lauren had written was a lot more complicated than I'd previously thought. Or it's a lot more, maybe complicated is the wrong word, but it was a lot more precise in terms of there was like a different loss function for, it's a 2-layer network, but there's a different loss function for each one. And they don't have like the exact same kind of terms in them. When you have Bayesian deep learning, or generally just like a Bayesian setting, it can be hard to compute the things exactly. And so you normally have to use approximate methods, especially for kind of high sample size or high dimensionality data. We have a low sample size but a high dimensionality. And so just spent like a week or so trying to understand how that works. The initial model that it had been working on was like very high sample size, very high number of features, like millions of features, hundreds, thousands, tens of thousands of samples, whereas I have like 150 samples odd and then around 2,000 features. So this model that my supervisor had developed had initially come from genetics, and I was just trying to sample it out. That's a bad choice of words. I was just trying to test it out. What I figured out over the course of the week was that it's a pretty good way to figure out whether you understand the model if you can implement it correctly. And it just made me realize I didn't understand this model at all, because it took me a fair bit of time to get it working, like a week or two at least. It was like, it's probably more like 3 or 4 weeks, because I kind of had this going even while we were getting the data set up. But I had like a simple Bayesian neural network with normal priors on each of the weights set up. But we're trying to use this thing called a spike and slab prior. The point of that is for our application, it's for more like feature selection. So you have a sparse neural network which has some structure related to the biological units in question. So for genetics, it would be, you'd have SNPs in your initial layer, and then you have genes, so like groups of SNPs are on a gene. For my work that I'm doing, it's proteins. So we're looking at like, you've got proteins in the first layer, and then you've got like groups of proteins, which is the pathways, and trying to just wrap that together. So that's increasing in order of biological units, and the sparsity kind of defines those connections loosely. And then you have this spike and slab prior to learn which are the important features per layer and per connection. It just made me kind of realize like it's not that simple to kind of implement this, because the issue with the spike and slab prior is essentially you have the normal distribution, and you can have a 0. So it would be like pi times the normal distribution plus 1 minus pi times the delta, or a 0. And because that pi is basically either a 0 or 1, And so you draw it from a Bernoulli distribution, a categorical distribution, which either gives you 0 or 1. And that just makes it very weird with deep learning, because deep learning requires things to be continuous. It's nice to have a bit more of a focus. Like the first week I was kind of writing up a few other papers that I had. It's hard to kind of get into the flow of things as much, and as well as dealing with like all of this kind of US admin, sorting my passports out, sorting the banks out. But I kind of feel like I've hit a nice stage. I'm going to be in New York for the latter half of this week, and then after that back to the grind for the last 5 weeks. And yeah, looking forward to it.